Thank you so very much, Professor Mitsumori. And good day to everybody here. It's a great honor for me to share this presentation with you across the globe. I noticed that close to 200 participants are joining today. So it really is a great honor. The COVID-19 cannot separately us out freely, right? So it's a great honor here to come back again to see everybody. I wish to express my sincere thanks to the organizer of this symposium, the third international symposium of the Korean Society of Rumen Function Studies, which is jointly organized by Japan and Korea. And the invitation to me today is feeding interventions to increase rumen fermentation efficiency and mitigate methane production. As we have seen very clearly, and it has been reiterated, that the enteric fermentation has significantly contributed to the global warming, why we have to take good care of the enteric fermentation, especially efficiency of fermentation, why we can mitigate the methane production, which is very important parameters and has a lot of factors associated with the mitigation factors. May I bring you closely to the scenario of feeding systems, which can really greatly impact on the mitigation and the efficiency of the production. Firstly, look at the feed resources, which is very important where you are, that what is available for the stakeholders, especially the farmers. Here in reality, you have to look at the availability and diversification of the feed resources in all sources at the gate, particularly on farms, especially those conventional feed resources, and together with the crop residues available seasonally. On top of that, the availability of the industrial byproducts, the core products, which have been available in each location, together with the introduction or interventions of the feed resources that can be established, which have been established already, but not fully be exploited, should be identified and make priorities in terms of which would be most prevalent and more efficient in dealing with the feeding systems, and onward into how can we improve the nutritive values or the quality of the feeds given to all classes of the animals, particularly with the roughages. We need to look at what and how we can improve the quality of the fibers, because the fibers is mostly utilized and exploited by the ruminants, right? So we need to improve either singly or integrated with the methods available, especially the simplicity and the cost effective of such methods, and onward into taking into account of the feeding systems which have been practiced by the stakeholders, either on farms or at industrial levels. So these feeding systems can have a great impact on how the feeds can be formulated or employed into the formulations in order to optimize utilizations of the rations given to the animals. And then onward into looking at the obtainable outcomes, which will be most efficient, in terms of quantity and quality, especially we're talking a lot about the functional foods that have been important for the consumers, particularly the functional protein, animal-based products. So all of these have to be taken into account and make prioritization into the feeding systems and making into the interventions for the stakeholders. May I just bring you into the impact of the rumen ecology, especially the pH phenomenon that has greatly impact on the fermentation efficiencies, which could be associated with the consortiums of microbiomes, which is reflecting on the functional roles of the microbiomes, especially those cellulitic bacteria, onward to other kinds of microbiomes available in the rumen, which will finally impact on the efficiency of the fermentation. So we have to understand and maintain the importance and the possibilities in buffering the rumen pH in order to maintain or improve the efficiency resulted from the utilizations of the feeding systems and the feed formulations and the ingredients offered to the ruminants, right? In particular, the range of pH that has been clearly reiterated that it should be, not lower than 6. Otherwise, acidosis may be prevalent, and then you have to build in other formulations to buffer back the rumen pH in order to effectively allow the cellulitic bacteria to do their good jobs. Looking at this consortium, you can see that there are quite complexities of the rumen ecologies that have been knowing already, and it would be the effective means of opportunities to capture the possible fermentation efficiency, especially in the consortium of the important species of microbiomes, as I have just mentioned. And then onward into looking at the fermentation that will impact on the methanogenesis that we are looking at here. And I thank my good friend here, Professor Atwood, for allowing me to use this diagram to show the methanogenesis that is reflecting the complexity of the methanogenesis of good and important species of the methanogens together with other microbes that can yield the fermentation byproducts. You can see that there are many groups that have interested in tackling methanogens, how they can reside effectively, and how can they be differentiated, and then what would be the reflections of the overall phenomenon of methanogens, right? Especially impacting on the final fermentation byproducts, especially the reflecting on the final quantity and quality of the byproducts. So it is very important area that we need to tackling more, especially employing the biotechnologies under the interest of the young scientists that have been showing a great way to tackling this phenomenon. Maybe I just point out to you, in terms of rumen characteristics that will eventually impact on efficiencies of the methanogenesis and onward to the efficiency of the fermentation byproducts. In particular, the ability to buffer the rumen pH, which is very important because this could be resulted by the feed resources and the formulations offered to the animals. And then onward to trigger the fermentation onward to the final fermentation end product, particularly the short chain fatty acids produced, especially C3 and the ratio of the C2 to C3, which will impact on the fat synthesis and the quality of the fatty acids in the meat and milk eventually, right? And the ability of the utilization of the hydrogen sink in the rumen is very important because when it's available there, as you can see, that how can you trap effectively to onward, utilize it onward for the synthesis of propionic acid, and then onward to other fermentation like nitrogen fermentation that will create a more efficiency of nitrogen synthesis, which is a coupling channel with the carbon utilization or carbon production, especially resulted from hydrogen sink, right? And as I have mentioned here already, that then overall here within the fermentation, the rate of fermentation, the degradability of the nutrients as a whole will eventually impact on the efficient synthesis of the nutrients, which would be liberated, that will be transported, forward into the lower gut for further synthesis. Again, here to show you the relationship of the fermentation in the rumen, okay, and the production of, in particular, milk and meat. When we mitigate the methane or reduce the synthesis of the trap of hydrogen for further synthesis of methane, we should be able to increase C3 and increase the ratio of C2 to C3, which means that efficiency is very important. We cannot just mitigate methane without obtaining a final product, okay, with the volume production of C3 and onward to synthesis of the products, as I just mentioned, in particularly the product of milk and meat, okay, growth and meat, which will be very important. What I have said that is that this should be disseminated for the understanding of the stakeholders. Here in Thailand, we have been working with many organizations and the farmers groups under the obligation and management of the Department of Livestock Development and the Dairy Farming Organization of Thailand. I mean, we have found, and it's very important to know that the farmers are not, keenly interested only mitigating the methane, right? They would be interested in the efficiency. How would they get, the output of meat and milk, better and onward to the sale of the products? So this is in practice, and it's very important and it's very relevant, to transform this technology to the users especially the farmers. I will reiterate a little bit more later on this. See, this is more practical in a way, especially in the tropics, because farming of livestock is under the hands of the farmers. And this is the majority of smallholder farmers or groups of farmers who have been really waiting for the recommendations in the, especially in the tropics. May I just mention, I also have a chance to work in China, and we are very lucky to, employ and transform this technology to the farmers groups by intervening the possible technology of feeding systems, making available feed resources available on top of what the farmers have been doing. So may I just show you a little bit on this feeding system. You can see here, this is called the food feed system, whereby we could intervene the combinations of the feed resources, starting from the fodders. The fodder trees can be a variety. For example, this first fodder tree or shrub could be Lucina here, could be planted around the farm, hedges. And second source could be like Flemingia, which is also available, and onward to Moringa. So 1, 2, 3 available shrubs could be planted on farm that could be harvested seasonally, whenever the farmers need it for feeding. Let's just enrich with a little bit of water here. Onward to look at grasses available. There are two kinds that the farmers have known of. The climbing type could be planted, and the erected type of grass could be, because with a small area of land, the grass could be best harvested from the erected type of grass, and the growth will be, subsequently produced. And onward to the climbing type of grass like Rusi and Signal, which is commonly grown in the tropics here, that could be grazed or can be cut carry into the pens. Here, this is simple kinds of grasses and fodder trees and shrubs, whereby the shrub of forests contain high protein, high level of protein, and very intermediate level of fiber. And on top of that, important is the secondary plant compounds of phytonutrients, as mentioned by the previous speakers, of condensed tannin, which is available, throughout the resources here. That could be utilized effectively into the feeding system. And then into the plot area, small area here, two cash crops or two economical crop could be intervened, like cassava, which is simply grown here in the tropics, here together with other legumes. Any legumes could be intercropped here, and the farmers can manage very effectively by harvesting the cassava here. That you can pick up the old leaves, sun dry or fresh, fed, as a protein sources, and together with the plant secondary compound available in the cassava naturally, and that could be managed if the feeding systems, feeding technology can be intervened through trainings of the farmers group. So this is a very good example that have been launched actually in the tropics, like in China, in Cambodia, may I say, through the EU projects, and Vietnam and Laos, as an example. That could be further intervened. Okay, onward to here is showing the good example, real example of the intervention of the feeding sources. Cassava intervened with Lucina and sunhelm and legumes. Flemingia shrubs can be available. Here, this is intervention of the grass, with the grass, and legume sources that could enhance the level of feeding in terms of both carbohydrate and also protein. Here is the flamingia. Its flamingia could be simply launched, cultivated here, and you can cut after like 4 months, and the regrowth would be harvested in the biomass rich in protein and very good level of fiber in terms of NDF here. It can be effectively utilized. Okay, this could be manually harvested. Okay, you can see good example the work in Indonesia of variety of shrubs, Lucina, starting from Lucina to Banya. You can see high level of fruit protein here on the top here, and some lignin, as I said, some plant-centric compound available here, and that could be managed well for feeding. Okay, sorry. Oops. Now, back to the benefit of using the fiber and the concentrate. As the late Oskov has presented very well that, reducing ratio of roughages to concentrate will effectively end up with the high level of C3 production and reducing the C2 production. Together with our work here, similarly, we received similar result here in terms of volatile fatty acid production profiles in terms of having high level of roughages and whereby reducing the concentrate, reducing the roughage to concentrate increase will end up having more propionic acid and less acetic acid, right? Okay. Now, this level of volatile fatty acid will impact further on the pH of the rumen. We would like to buffer it up to 6.1 in order to get the cellulitic microbiome effectively utilize their substrates, right? So to do this, you can see the diurnal fermentation, which affect on the pH here. You can get a high level of concentrate. You will then eventually affect the fermentation in product and pH. Lifting it up with the roughage, you will see pH buffer up, which is very important to maintain the pH. Okay, how you do that? You have to move forward. Using the rumen enhancers, especially employing more plant secondary compounds or phytonutrients, which have been addressed by the previous speakers already that will then impact the fermentation. Now, under our umbrella and our approach, we would see what would be most relevant and more practical to utilize plant secondary compounds or phytonutrient sources. We see that in the tropics, we have a lot of tropical foods, and their peels contain high level of phytonutrients, particularly mangosteen, rambutan, or golden fruit, particularly mangosteen peel. Okay, and may I mention rambutan, and then onward to papayas. So we can see what concentration of the plant secondary compounds were available, and what combination we can tailor down into the pellet, so we can then supplement more effectively. So may I just show you with further work that we have done here. You can see what can be beneficial effect of using the phytonutrients, right? You can see that this phytonutrient would interfere with the microbiome cell membrane that will affect on the transport of the nutrients or the ions that will then affect on their residency and their effective activities of fermentation. The phytonutrients will also directly block the metabolic functions of the microbiome, particularly the bacteria in their secretion of the enzymes and or activation of the enzyme into degrading the substrates. Okay, and also they could prevent development of the bacteria structure like flagella, which is essentially required for the adhesion to the substrates. And this bacteria, particularly the cellulitic bacteria, for some species would be affected. And most importantly is the impact on the protozoa population, okay, expansion living in the rumen ecology that could be definated, whereby the adhere methanogens, as we all know that they adhere to the surface of the protozoa, that will be eventually be mitigated, that will be eventually be reduced from the rumen, thereby the methanogenesis will be also mitigated. And we have also found that the effect of phytonutrients has impacted on Fibrobacter succinogenes, which is very important. It's a non-hydrogen producing bacteria that should receive more research and see what kind of dose of phytonutrients that could be effectively impact on Fibrobacter succinogenes, which is very important for the ruminants. You can see here, these peels, food peels, could be combined together here and then work both in vitro and in vivo. And under this presentation, I'm very lucky to present to you more in vivo work to affect the impact, final impact of the fermentation of this food peels pellet containing phytonutrients. Sorry. Here we go. This is an example of the product, one product that we formulated using banana peels called BAFLOP. You can see the level or dose of supplementation from 0 to 3% of dry matter intake would greatly impact on the rumen pH from 0 hour to 0.6 to hour 6. And you can see they will buffer up the rumen pH as the level of supplementation increase. The rumen pH will parallelly, and here is the average values of the hours. You can see very clearly as the dose increase, the rumen pH subsequently increase. Okay, and another work here, looking at the fermentation and products of using mangosteen peel, okay, at the lower level at 100 grams per head per day. Okay, another source, banana flour, and combination of the two. And you can see here, mangosteen peel and the combination would be greatly impacted as compared to the control and also only the mangosteen peel. You can see here the banana peel and the combination of banana and mangosteen peel would better produce more volatile fatty acid and more propionic acid in the rumen. Another work with dairy cattle, we employed the rambutan pellet together with some urea. You can see these treatments of it. Okay, there are 4 treatments here. You can see the milk yield and the 1.5% fat-corrected milk here were significantly improved by the supplementation of the rambutan food pure pellet and the combination of rambutan pure pellet and urea. Especially urea as a source of non-protein nitrogen would also additionally improved when we fed the rambutan pure pellet. This is very important for the production. As we can see that when we supplement this source of phytonutrients, as I mentioned, the farmers would be more interested in seeing the final impact on their milk yield. Look at pH again. To another experiment, 5% body weight concentrate supplemented with MARABAC compared with the high level of supplementation of concentrate at 1.5% and supplemented with the same level of MARABAC. You will see the rumen pH here was buffed up after 24 hours. You will see all the pH was buffered up both at the lower level of supplement and at the high level, particularly at the high level of supplement. Here you will see because the pH was dropped down, and then when fed with MARABAC, it was up in the rumen. What about the volatile fatty acids? You can see here, propionic acid was significantly improved with the supplementation at the low level of MARABAC and also at the high level, significantly improved, as well as the ratio of the C2 to C3. While the methane production was dropped significantly from the control or the low level of supplementation. You will see here on these two treatments. So the supplementation of the pellet containing phytonutrients was significantly improved. Here, may I summarize and present to you that the in vivo work currently obtained through using a variety of shrub leaves and food peels pellet in various species of ruminants, from beef cattle, swamp buffalo, and onward to dairy cattle. You will see their impact on the fermentation, particularly improving the C3 production, okay, and reducing protozoa. All of them have impacted on reduction of the protozoa population, and the volume methane production in the rumen were also significantly reduced. These were all in vivo work using the supplement pellets of various sources that I have mentioned. Onward to others, shrub leaves and leguminous tree leaves like tamarind, together with like herb sources, bamboo grass pellet. This is from the leaf, Tiliacora species of bamboo. We call the common name is bamboo grass here, which contains high level of phytonutrient. Okay, have shown similar impact on the rumen fermentation ecology, particularly the enhancement of C3, the mitigation of of methane, and the reduction of the protozoa population, which have a very interesting result and a high promising potential effect that should be developed further in terms of building it more with regards to the dose of supplementation and the production in terms of quantity and quality of the products, particularly the functional food, functional protein from either peptides or amino acids, as well as the amino acid and peptides, as well as the fatty acids, especially unsaturated fatty acid of CLA as important. And now a little bit with the regulation of the government. As I mentioned, that we have been working closely with groups of farmers and departments who closely associated with the groups of farmers like Department of Livestock Development and also the Dairy Farming Organization of Thailand, that we could closely contact with the farmers and transform these research findings. But under the current scheme of the government through the BCG, bioeconomy, circular economy, and the green economy, that the government has launched not only the research for development grants for the researchers, but also the on-site work, particularly through the sector of food and agriculture that currently have been launched in Thailand to support and promote the interest, and providing financial support for the researchers throughout the country. And we call this as an area-based development through, all sectors of development here. And the government have launched these schemes and also making available the final budget. Before I end my presentation, because the time is short here, may I just show that I'm very lucky and happy to be here talking to you guys here, and which bring me to my friends, Professor Kobayashi and Professor Ha, a long time ago together here, and others, many, you can see over here, friends from England, So it's a great opportunity for us to get together more and more. And lastly, may I just conclude that throughout this presentation, the work in the rumen is very important, especially the factor of pH that will eventually affect on the fermentation, particularly the methane production or the utilization and the association of the microbiome worked over here. And available feeding system, diversity of feed resources will have a great impact on the practical feeding systems, especially geared up by the stakeholders. Not only the farmers, but the industrial level should also think that this is practical, this is very important, and this could be cost effective for the stakeholder, for the farmers, because when they reduce methane, industry should spell out clearly that this will eventually affect on high level of benefit, selling their products like meat and milk, right? Development of products, simple and more easily intervene, like using food waste or food peels containing phytonutrients like this, could be very important, and there's a future opportunity for industry to help develop and make it more practical and more available for the end users. And again, even with those results obtained already, more research should be geared up and should be more warranted in order to investigate and to obtain more data, containing on their effect to quality and quantity of the products in ruminant, particularly the functional nutrients available in the products, as I have mentioned here. And I believe that dietary manipulation here, among other schemes as presented by other previous speakers, is also has a future of interest and is very promising to explore even further. With this, may I thank all the audience and my friends, speakers here, to meet each other and also to share the information. And we look forward to sharing even more and more in the future. Thank you very much. สวัสดีครับ